Hello, everyone, and welcome to the discussion today. My name is Chad Schwartz, and I'm a Senior Manager of Global Product Management within Thermo Fisher's Cell Culture Group, responsible for management of the proprietary media formulation product lines. Today, we'll be providing an overview of some of the common challenges associated with cell culture media manufacturing, as well as some best practices and considerations as processes scale from bench to the manufacturing suite. However, this discussion simply cannot overview all considerations, and in fact, much of the rigor associated with process development and technical transfers will not be able to be reviewed today. On the agenda for today will be a comprehensive review of the considerations for scaling the cell culture process from the bench to the bioreactor, and we'll first start by introducing some of the challenges of the day. We'll then move into the importance of ensuring quality, deciding upon the preferred media format to use, the benefits of qualifying at small scale using non-GMP pilot scale manufacturing, and finally, we'll end the discussion today with some of the key components to look for in a media supplier as your drug moves through its development journey. As with all industries, there is the drive to do more with less. Maximizing efficiency leads to a decrease in cost of goods, allows pharma to keep up with the growing demand, and ultimately leads to a reduced price for consumers. Optimizing protein and vaccine development and manufacturing is key to enabling the speed and success rate by which life-changing drugs enter the commercial market. Very often, scaling up is one of the most difficult parts of the drug development journey. We'll review some of the common challenges, identify the risks associated with taking certain paths, and then review some opportunities to improve process. So what does scaling up actually mean? Here we are referencing a transition from the small-scale development work and process qualification to the large-scale manufacturing of the biologic once commercial production is ready or required. The time at which we typically see this transition will vary from customer to customer, but it's usually immediately following the preclinical evaluation. Today, we're just going to focus on the upstream portion of the biologic manufacturing, specifically the utilization of cell culture media used to manufacture a biologic of interest. So what are some of the considerations with regard to cell culture process? Firstly, the media composition and format is critical to consider. Most process development work is done in volumes under 50 liters and performed typically using the liquid format. For commercial production, however, dry powder format is preferred due to the costs associated with shipping large volumes of liquid, and scales can be as large as 50,000 liters or even more. Thus, it's important that your media formulation is compatible with a dry powdered format. The next consideration is related to performance. Essentially, it is very important that the titer and other key process output variables are as close to equivalent on the large scale as you would see in the small scale or on the bench. The next category that should be considered centers around the raw materials of the formulation. Not all raw materials are able to be sourced at large scale in the same fashion or ease as you would see at small scale. The last thing a manufacturer would want to realize is that after all the hard work in qualifying their media formulation in early stages with a particular formulation, that would need to be reworked since one or two of the media raw materials cannot be procured in sufficient quantities to meet commercial volumes. Finally, I'll mention that there are further requirements that one must consider during the transitory period, such as equipment qualifications and validations, setting specs on the finished good to prevent variability, the packaging and shipment requirements, sometimes including temperature monitoring or otherwise, and how that material will be received at the site of manufacture, as well as many other items to ensure that the process is streamlined once commercial production can be green-lighted. As a deeper dive here, we'll highlight two of the challenges a bit further since they are so important. As mentioned on the previous slide, the makeup of the media formulation is critical, as supplier shortages or shutdowns can lead to significant production issues. Further, low quality raw materials can cause a variety of issues and lead to variability or poor performance. Both of these are issues typically which lead to rework that can be extremely costly and impactful to a company's commercialization plans. Often, variability is more present in certain raw materials in non-established markets, which means it is very important to know the source of your raw materials. To screen vendors, many organizations employ a multi-level process of vendor qualification. These type of screening processes can include comprehensive vendor surveys, multiple batch testing, audits, ongoing monitoring, and scoring system. Other common issues seen during scale-up can include significant unforeseen costs. For example, going from research use only to CGMP requires more rigor in all aspects, and many companies are surprised to see the difference in costs between the stages and/or the grades of material. Finally, one other common issue to highlight here is around space constraints, inventory control, and media shelf life. Cold room space is often at a premium, and if not available on site, many companies will resort to third-party storage solutions that can increase the complexity of distribution, the use of that material, as well as ultimately raise costs. So now that we've discussed the challenges, let's move into some of the solutions. The first focus area is going to be around quality. From a cell culture media perspective, a supplier with a global footprint and robust equivalency program can supply continuity and confidence that the media of choice will arrive on time, in full, repeatedly over and over, with minimal defects or performance complaints. Ensuring consistent quality in media manufacturing starts with robust quality management procedures, followed by strict raw material sourcing, as well as the characterization of those raw materials and finished good compliance. Partnering with a supplier that has robust quality procedures helps to mitigate risk during the scale-up process. Some of the questions you may ask your suppliers include how they reduce risk of cross-contamination at their facilities. Are all of their facilities ISO certified, and how often is that site audited? Biologic manufacturers should also ask to be taken through the complaint management process, or really any other documented control process that may be applicable to their process. Finally, it's important that manufacturers feel comfortable with their suppliers, so frequent site visits, audits, and even technical discussions with the supplier's quality team is really critical to ensure future success at scale-up. Cell culture process can be highly impacted by the influence of certain metals or contaminants, which are most often derived from the purity of the raw materials within the media formulation itself. Many global media suppliers implement raw material control strategies which help to maintain consistent quality of the finished good. Some of the same questions that we asked on the previous slide should be considered here, but more specifically, it's important to understand how the vendor qualifies their vendors and what standards or specifications are implemented to reject vendors or specific lots from these vendors. As mentioned, the country of origin of the raw material should be considered and whether multiple lot batch testing is required to onboard a new vendor into their quality management system. Finally, change notification programs should be established by your cell culture media vendor to notify of any changes to processes, vendors, or even specifications from those vendors. As briefly mentioned on that previous slide, variances in trace elements or metals, typically heavy metals, can cause big challenges in cell culture performance. As shown here, the elements which cause the most significant change or detriment to cell culture performance include copper, zinc, manganese, iron, selenium, as I mentioned, many heavy metals. Iron salts, such as ferric citrate or others, appear to contain the highest number of unexpected elements and should be limited within the media formulation or at least strictly controlled or monitored. It's important to understand these details during the scale-up process, and working with a vendor with a proper subject matter expertise in cell culture media is pivotal so that there are no pitfalls along the way. At Thermo Fisher, analytical capabilities to test for elemental impurities such as ICP-MS have been developed as well as validated across our global manufacturing sites to proactively test our raw materials and finished good cell culture media. Critical raw materials are identified and continuously monitored to understand baseline impurity levels. Further, supply chain variability in raw materials is controlled as well as mitigated using stringent risk assessments, and traceability of cell culture media for lot-to-lot variability is tracked using a trace element database, as shown here. During the scale-up process, there are sometimes solutions that actually present themselves that can enhance productivity. For cultured media, this discussion could include media formulation optimization, implementing certain supplements or concentrates to improve the process, or even changing the packaging to better suit the production on the manufacturing floor. However, we'll dive a bit deeper here into the most common conversation, which is around the format of the media. When scaling, an additional consideration is selecting a media format that will best achieve your goals. Typically, media can come in either dry or liquid format, and each format has different benefits or disadvantages. For instance, liquid media is easy and ready to use, requiring no reconstitution. This helps to reduce the risk of cross-contamination and helps to improve process efficiencies. However, liquid is quite expensive to ship around the globe and quite cumbersome to store, typically at 4 degrees Celsius. Further, liquid media is typically less stable than its dry powder counterpart and thus has a shorter shelf life. Dry powder format, on the other hand, is much more economical in terms of shipments and can typically be stored at room temperature, which alleviates space constraints in walk-in refrigerators or other 4-degree spaces. However, reconstitution requires a separate reconstitution liquid tank, adds risk contamination, and can be quite time and labor intensive. Thermo Fisher's proprietary advanced granulation technology, or AGT for short, this format takes the advantages of traditional dry powder format but it includes additional benefits such as simple reconstitution. AGT is a crystalline dry format which speeds up dissolution times, reduces inhalation risk, and is auto-pH'd, which reduces the potential for user error. Some additional factors to consider before deciding on which media format to go with include the resources required for media preparation, the availability of in-house storage space, costs associated with shipments, and then finally an analysis of required quantities and their associated overall costs for shipping and storage. Often it's discovered that as processes are scaled, it becomes much more economical and practical to leverage a dry powdered or advanced dry powdered format, such as AGT, versus liquid, despite the inherent advantages to liquid. Regardless of the format that is chosen, following those considerations above, it is critical that your supplier offers suitable batch sizes of high quality products and the choice and flexibility of format packaging to match your processes in-house. It's important that during scale-up, procurement is working closely with the material sciences and the manufacturing teams to ensure compatibility between the vendor products and the manufacturing process. Further, during this transition from bench to manufacturing, it is critical that your supplier has a wealth of cell culture media expertise, as well as a suite of custom services. From qualification support to formulation development, partnering with a transparent and expert supplier is key.
In this next section, we'll take a closer look at small-scale, non-GMP manufacturing, including the benefits of prototyping, our Thermo Fisher network of pilot labs, and a case study on dry powder media scale-up considerations. One of the most pivotal steps prior to scale-up is media qualification at small to mid-size scale. Since this qualification does not typically require GMP media, many large multinational suppliers offer some version of small-scale test batches. This is highly recommended for a quick and cost-effective way to ensure the material and supplier meet your expectations prior to proceeding with full-scale commercial production. Further benefits on the media manufacturing side of things is that it allows for a feasibility scientist to confirm the manufacturability of your formulation and begin to create batch records, which can be easily transferred to the GMP work center once you're ready for large orders. To reiterate, the rapid prototyping non-GMP media option is one way to bridge the gap from preclinical to late stage. Thermo Fisher's GIBCO Rapid Prototyping Program utilizes CGMP qualified raw materials and processes, which again makes the transition to GMP seamless. This program allows biologics manufacturers to evaluate raw material formats and modify existing formulations by adding novel components or reducing and removing components that do not drive cell culture performance. Additionally, Thermo Fisher's GIBCO Rapid Prototyping uses the same raw materials as you would get in GMP, so you can source and test unique raw materials at pilot scale. Further, even the manufacturing processes mirror that of our CGMP production, so a prototyping approach can help to accelerate and simplify technology transfer during scale-up. Looking at GIBCO Rapid Prototyping's footprint, 2 labs are located in North America, with the legacy Grand Island, New York site, as well as Hunt Valley, Maryland, respectively. More recently, we've opened a new piloting facility in Suzhou, China, with the intent of having a manufacturing site in China for China. In preparation of opening this facility, we've extended our supply chains and harmonized the processes so that our North American sites and our site in China align with our GMP facilities. At Thermo Fisher, The rapid prototyping teams are highly technical and dedicated to helping customers with their small-scale pilot needs to instill confidence in production scale-up. The GIBCO Rapid Prototyping Labs focus on a 2-week and 4-week lead time adherence, so that rapidly provides material to do your testing and evaluation. When we look at our different sites across the network, our Grand Island site is our legacy site and has capabilities for liquid media, and liquid concentrates up to 600 liters, while our Hunt Valley and Chuzhou sites have liquid capabilities to 200 liters. For dry powder media, our sites are equipped with FITS mill and pin mill capabilities, where Grand Island has a 10 kilo max batch size, and Hunt Valley and Chuzhou can leverage up to 30 kilo batches with FITS mill and pin mill. For advanced granulation technology, Grand Island is our flagship site with capacities up to 8 kilos as well as a 4-week turnaround time. All of our lead times are based on having raw material and component availability in scenarios where there's custom packaging and special order chemicals required that may extend lead times and require longer development timelines. Next, we'll take a look at a dry powder media case study. This case study is based off a 2-phase strategy using complex DPM formulas to deliver rapid prototyping material and help sustain quality with scale-up. In phase 1, the initial non-GMP lots were produced by GIBCO Rapid Prototyping teams. Standard testing included pH, osmo, and additional testing was requested for analytical work for bioburden, endotoxin, glucose, salts, amino acids, water-soluble vitamins, and trace metals. For phase 2, an internal tech transfer and scale-up took place and was supported by the GIBCO cGMP media manufacturing services and included several large-scale lots of the same formulations. Testing of DPM evaluated for equivalency between GRP and the cGMP produced material. Case study results. Overall, the comparative GRP to cGMP test analysis showed acceptable component group level equivalency from small scale to large scale cGMP productions. The test results for bioburden, endotoxin, and the individual analytes were all within established specification ranges. As shown in the figures below, test results for pH, osmo, amino acids, vitamins, and trace metals are all within established spec ranges. To summarize the case study, utilizing this 2-phase multi-lot strategy can help deliver the required rapid prototype material and data analysis needed to ensure that consistent product quality is maintained from development or transferred through scale-up to cGMP. The success of this 2-phase multiple lot strategy is further supported by detailed formulation batch records and well-established technology transfer processes, harmonization and common sourcing of GRP and cGMP qualified raw materials, use of identical or equivalent equipment design and manufacturers, robust raw material and quality control systems, control and quality systems in place with each site, and site-to-site manufacturing comparability from well-established site and equipment validation processes. We will now transition to Chad to talk more about supply assurance.
The last consideration that we'll touch on today during scale-up is the one that is most often overlooked. After considering both the quality management system of the supplier as well as evaluating the media and supplier for technical performance, reliability of the supplier needs to be assessed. There are many reasons one might choose to outsource their media manufacturing. Some of these may include to improve productivity and operational efficiency, to reduce risk by sourcing raw materials through a qualified supplier, or maybe to increase access to capacity, expertise, and the facilities that perhaps the customer does not have in-house. Some of the things that you can evaluate when considering to outsource your cell culture media include the segmentation of animal origin and animal origin-free facilities, whether that company has a global footprint to maintain redundancy, capacity, and supply continuity, and then finally, whether or not that supplier has a world-class logistics and distribution network that really help to streamline supply as the formulation is commercialized. By partnering with a preferred media vendor, again, as processes are scaled here, you are ensuring a consistent and harmonized supply source, which helps reduce costs, increase productivity, and mitigate supply risk. Suppliers usually take advantage of multiple large facilities that can produce required volumes in desired formats. Batch sizes are a very important consideration for cost, and having equipment that matches the required customer sizes is critical to meet expectations. It's also important here that these facilities are placed strategically across the globe for logistical purposes. Finally, in terms of redundancy, having secondary or even tertiary sources for critical raw materials is essential for manufacturing suppliers for more guaranteed supply. At Thermo Fisher, we are positioned globally. We can offer between an 8 to 12 week turnaround time from order to shipment, and we provide media for batch, fed batch, as well as perfusion applications using platforms of mammalian and microbial nature. We do offer multiple formats, liquid concentrates, your traditional dry powder, and then that innovative advanced granulation technology format. Thermo Fisher's global footprint and redundancy throughout the world ensures a consistent supply. All major manufacturing sites are ISO 13485, 21 CFR 820 certified, and registered with the FDA. Thermo's major manufacturing sites include Grand Island, New York, which is the birthplace of Gibco, Paisley, Scotland, and Miami, Florida. We employ a robust harmonization strategy amongst these three major GMP manufacturing sites, which include harmonizing the raw material supply, harmonizing those manufacturing environments, and testing for product homogeneity under the same QC tests. Additionally, we've harmonized our processes from blending to milling to dispensing, etc. Our processes are harmonized across all three major manufacturing sites. Finally, from a quality standpoint, all of the batch records and procedures are under a central quality management system, and our QC release testing is validated under that same quality management system. In addition to Thermo Fisher's redundant harmonized sites, we also employ a multi-layered business continuity and risk mitigation plan to help supply continuity for our partners. This includes an emergency response plan, backup power of critical environmental systems, and customer notification program for changes in any product specification or otherwise. We view our customer relationships as a partnership, and we pride ourselves on transparency with regards to our processes and products. To overcome barriers in biologic development, there are three areas to prioritize. Firstly, raw material quality and consistency needs, which include user requirements, the level of regulatory compliance and risk assessment of the supply chain. Secondly, knowing in advance what factors will streamline the scale-up process, such as what raw material attributes will facilitate this phase in supply continuity. And then finally, a proactive approach to outsourcing and collaboration with your supplier throughout the process, like considering pilot scale material, GRP, for example, and other customization options to set up your system for success at scale up. As you research potential collaborations, you'll find that Thermo Fisher is, of course, one strong option to consider, from biologic development all the way through commercial manufacturing. We're proud of our worldwide leadership in this area and encourage you to check out our web section to learn more about the strengths and experience that we bring to the table. The site also has a convenient information form where you can ask specific questions on your particular needs, and we are happy to respond accordingly. Thanks again for attending our discussion today on the scalability considerations in the development and commercialization of biologics, and we look forward to discussing with you in the future.
Alrighty, Chad, Rob, this was a great presentation. Just everybody, Miles Skoda here. I'm your host and moderator for the event, and I'm going to get us into Q&A here in just a second. But let's just a couple reminders. If you do have a question, please submit that at the bottom of your screen. There should be a box for the Q&A with a prompt from me asking to submit your questions there. And then if you did want to reach out to our speakers independently, their information should be kind of on the left side of your screen. There should be a little, or on the right side of your screen, there should be a little email icon. Click on that and contact them directly. And then finally, we've got some additional content we've kind of curated for you on today's topic. I think 5 pieces, bottom left side of the screen, just related to what everything was covered by Rob and Chad today. And so with that, let's get into this Q&A. We've got certainly a few questions here to go through. But definitely make sure to submit your questions. We'll get to as many as we can here in the time we have. So guys, the first one I have in here is someone's asking, what are the different analytical methods used to qualify raw materials besides ICP-MS for elemental impurities?
Thanks, Miles. This is Chad here. I'll take this one, Rob. I do think that ICP-MS is certainly the workhorse for trace element impurities. That is our typical analytical assays for controlling trace elements in all of the suite of raw materials that we have that go into our finished goods. There are a number of other analytical assays that we do employ for raw material control. Those include, LOD, specific rotation, and many other analytical tests. But ICP-MS is certainly the one that's used most predominantly for impurities.
Beautiful, thanks. Let's see, moving on to another one here. What kind of documentation is provided with the products made using your non-CGMP manufacturing service?
Hey, Miles. Yeah, that's a great question. Our GRP is considered a research use only intended use for our products. So from a documentation standpoint, we can provide a memorandum of assay is how we communicate any standard test results. So you get pH, osmo, insolubility, or sterility for liquid products. And as RUO, we typically wouldn't provide like a certificate of origin or any other documentation that you would get from our CGMP products. But for GRP, again, we have a memorandum of assay, and we'd also provide any type of recon instructions if you're doing a DPM or AGT.
All right, perfect. Thanks for that one. We've got a few more here. Again, audience, if you've got any more questions, definitely submit those. Should be able to get to a few more here for sure. Bottom of your screen, little Q&A box. Just type it in and let us know. Let's see. So this one, they're asking, can you describe the product testing available for media produced using, I think they mean the GIBCO, the rapid prototyping services versus CGMP manufacturing?
Yeah, I can take that one too, Rob. So I think Rob already explained, right? Our primary test that we use in the non-GMP, what we call GIBCO rapid prototyping labs, that is pH, osmolality, solubility, and sterility. Those are what comes in terms of the documentation to that former question. Within GMP, we really open up the suite of QC tests that are permissible on our certificates of analyses. Again, for demonstrating that we have successfully manufactured your products. Some of those additional QC tests could include endotoxin, bioburden. We can look at water-soluble vitamins, amino acids, even growth promotion assays, things like that. So the certificate of analysis and the QC tests that are performed on your finished goods are really customizable. Right? We allow you to customize the type of QC that you require for your media at Thermo Fisher. And we can provide some literature related to that full suite of QC tests as part of this as well. Thanks for the question.
Yeah, perfect. Great stuff, guys. Let's see. I think we can get a couple more in here. So up next, someone was asking, could you talk more about or review kind of the lead times and capacity sizes per format that you discussed?
Yeah, sure. So from a GRP perspective, we really strive for a 3 to 5 week lead time across our different formats. Our advanced granulation technology format does require a little bit more pre-work, we'll call it, in the GRP labs. So we do strive for, say, 6 weeks for AGT GRP productions. Obviously, this is all, again, contingent on having raw materials available, and our lead times do fluctuate over time, just given capacity and the volumes that are going through the labs and the different production lines that we do have. In GMP, our lead times are also highly variable. We are striving, and I think it had it on the slide there, 8 to 12 weeks in lead times. Again, the caveats that are mentioned with regards to having raw material availability and specific to the production line itself. But that is our goal, is 8 to 12 weeks in the GMP side of the business. And we've been able to accomplish that through investment in our capacity across our 3 global sites. So the Paisley, Scotland site, the Grand Island site, and the Miami, Florida site. I hope that answers the question.
I think it definitely does. Yeah, thanks. That was perfect. Let's see. So I think we're going to be able to fit one more in here. I have a couple questions here, a bit specific audience members, and so I'm going to save those for later, and I'll pass those along to Rob and Chad afterwards. But let's cover this one. They're asking, how do you ensure scalability between non-CGMP media manufacturing and GMP manufacturing?
Yeah, another good question. So with our GIBCO Rapid Prototyping Lab, we have a team of feasibility scientists that look at the formulation and really assess it for manufacturability. And when it's in our GRP, the batch records that are built, right, to manufacture and execute that specific formulation are easily transferable to our CGMP team. So when you're starting with GIBCO Rapid Prototyping, that helps ensure that scalability, because the way the batch record is built is easily transferable over to the CGMP side. And knowing that our equipment and processes align, it makes it, again, a seamless transition to go from GRP over to CGMP. So it really comes back to that, reviewing each formulation for its manufacturability and being able to successfully execute that. So when you're able to do a pilot batch in GRP and you can evaluate it on your end and make sure it works, it's, again, a seamless transition to go to CGMP.
Yeah, great answer, Rob.
Right.
The only thing I would add is, again, we've just recently published a white paper on that particular topic where we looked at a few different formulations, starting in GRP and then scaling to GMP. And, you know, that's a really nice asset that we have to demonstrate just exactly that question, is how we're ensuring the scalability and the equivalence amongst the non-GMP labs to the GMP labs. So happy to provide that as well as a takeaway from this webinar here.
Perfect. Yeah, Chad, Rob, thanks. I think this is actually going to be a great place to wrap up too. Just a notice to the audience. Again, we do have a lot of resources about today's topic. I think that white paper might be over there on the bottom left corner. If not, get in touch with Chad and Rob. Right side of your screen, a little blue circle has got an email icon inside of them. You can reach out to them directly right now and just, you know, let them know what you need. Let them know any other questions that you might have that are, you know, maybe too specific to ask here. But with that, again, Chad, Rob, thanks so much. This was a great presentation, a lot of really good insights and a lot of really good information, and I think everybody really appreciated. And audience members, thank you for joining us today. I appreciate your time and you being here as well. And so with that, we hope you all have a great rest of your day, and we'll see you next time. Bye, everybody.